Dear ladies and gentlemen, thank you for visiting my presentation with the title Micro-RNA Target Prediction of Avian Z-linked DMRT1 gene during sex determination in chicken. This study is presented in ICSAE-8 2021 with the paper ID 229. As introductions, I'm introduce myself. My name is Sigit Prastowo. I'm coming from Department of Animal Science, Faculty of Agriculture, Universitas 11 Maret. As we know that the sex determination in dimorphic animals, such as chicken, is controlled by the expression of double sex and MAP3 related transcription factor 1, or we can call here DMRT1. This gene is act as sex determination which switch the critical factor needed for testis differentiation and as agonist of ovarian development. As we can see here in the left figure, that sex determination is started from the fertilizations, and in chicken there is a two sex chromosome, Z and also W, and this is will determine the sex in chicken. And gene which is involved in chicken's gonadal sex determinations is for male, which having a double Z as a sex chromosome is the center is DMRT1. This DMRT1 expression will correlated with other genes expressions which determine the cell into testes, while in Z and W chromosome in female, there is the expressions of the DMRT1 is not relatively high compared with the male, and this determine the cell into ovary. Then DMRT1 here is very important to determine the testis or the development of cell into testis. As I mentioned before, that the DMRT1 gene is a transcription factor, and this transcription factor, as other gain, is its expressions is controlled by factors. One factor which control the gene expression is the micro-RNA. Micro-RNA is short nucleotide which consists 19 to 24 nucleotide, and its function is to fine-tune the gene's expressions. As we can see here that part of micro-RNA will attach to the micro-RNA, which can be overexpressions or direct overexpressions of the gene. One gene can be targeted to many micro-RNA or vice versa. This micro-RNA can modulate gene expression in specific port target. Therefore, this study aimed to predict the micro-RNA candidate targeted to DMRT1 expressions in chickens. This study using in silico method. We divided into two study methods. First, we start with the predictions of micro-RNA targeted to DMRT1, followed with the second method is using genes ontology analysis. In the first study method, we use in silico prediction using three bioinformatic database, namely micro-RDB, target scan, and also micro-TCDS, following the clustering where the common micro-RNA was found in at least two databases were selected as candidate. The selected candidate are the shortlist then processed to gene clustering into gene ontology analysis. In the gene ontology analysis, we employ the genes targeted by micro-RNA candidate using miRPath versions 3 as the software for geoanalysis, and we using the default setting of the software of p-value 0.05 and micro-3 were set micro-t with set in 0.8. The resulted heat map figure, and biological process, and 8 cluster microRNA were then retrieved. As the result here, the predictions of microRNA with my target to the MRT1, resulting 78 unique microRNA from 3 databases, as resumed in this table. So from target scans, there are 40 microRNA predicted, microDB 11, and micro3 CDS is 27. So in total is 78. And according to the clustering analysis using Venn diagram, we found 8 microRNA, which is at least in the 2 database, and we can select as the candidate. So one microRNA was clustered in database targets 10 and micro-RDB, namely GGA MIR 6701 3P. GGA is the abbreviation of gallus. Gallus means microRNA in chicken. Then one microRNA also predicted in the database of micro-RDB and micro-TCDS, and 6 microRNA in target scan and micro-TCDS. Following the selecting of microRNA candidate, we then input to the gene ontology analysis, and then from this 8 microRNA, there is a lot of target of microRNA here. For example, GGA microRNA 76701 3P is targeted to 473 genes, and from all these microRNA in the gene ontology analysis, we found a molecular functions here: intracellular, integral component of membrane, protein binding, nucleus, and cytoplasma, and also membrane here. The selected microRNA analysis also resulting heat map analysis, which is clustering the positions of the microRNA and the specific molecular functions or intracellular. I mean that's specific gene functions. And here we can see that there are 7 molecular functions here, 7 pathway here, which is controlled by this microRNA list. Then the radar of color here in the heat map shows that there is more influence or more degree of this microRNA to the specific functions here. According to the microRNA cluster dendrogram, we found 3 clusters of the microRNA, which is functions in 3 pathway here. This is for examples. There is a GGA-MIR 1580. There is one cluster here, different with the GGA-MIR 1382 3P here. And then also this is different with the 1, 2, 3, 4, 5, 6 microRNA cluster here. And then according to the pathway cluster dendrogram, we found specific for molecular functions, then 2 intracellular and integral component of membrane, followed with membrane, cytoplasm, protein binding, and nucleus as pathway cluster dendrogram. The cluster analysis using dendrogram, we have 3 pathway here, and this is so that this pathway shows a specific function for specific development in the cell development. As we know before that the MRT is a sex regulator for the determination of sex in gallus, and then this DMRT1 is a transcription factor that works as regulator for testis development, as we can see in the right figure here, that the DMRT will determine the differentiations of the cell into testis in conjunction with the function of SO89. And then this DMRT also reported in the previous study that it exhibits a gonad-specific and sexually dimorphic expression pattern to maintain the fit of or determine the further testes or ovary. Then the DMRT here involved in the cell development of sexual organ by control the cell growth. So there is a control of cell growth here in the differentiation process, or we call also this cell growth is proliferation in embryonal state. This is supported by the result of our previous GO analysis, which shows gene pathway related to cellular growth, cellular communications, and its interactions. So here, the pathway cluster dendrogram shows the several functions which support to the cell development or cell growth. Of the in silico study of the DMRT1 targeted to micro-RNA or vice versa, we also found here some report of GG micro-RNA in especially in the reproduction process of chickens. For example, here that GG-MIR1684A and 1434 are connected with the functions of chicken eggs production rate. Here also microRNA 34B, 34C, and 216B. This is control the proliferation, cell cycle, apoptosis, and metastasis, and expression of genes which is control in ovarian of high rate egg production in chickens. In male, microRNA 202 regulate testis development, and microRNA 215 determines sperm motility, as well as microRNA 155 and 74805B. According to this report, we can see there is a possibility functions of microRNA in chickens, which can be open the possibility to use microRNA for the future study, especially for functional study. If we can know the function of specific microRNA into specific target genes, then we can manipulate the microRNA actions to improve or decrease the gene expression, will lead to the biological functions. It also affects on the biological functions. Therefore, the use of microRNA in this study, especially in the chickens, will be promising to enhance the productivity either in local or commercial bird productions. As the conclusions here, that I8 microRNA, GGA microRNA as candidate targeted to DMRT1 genes, the geoanalysis reveals 7 distinct pathways of gene or biological functions of DMRT regulating during sex determination in chicken, with all these functions related to the cell proliferation process. Further study is required to validate those GG microRNA targeted to DMRT1 to exactly know the sex differentiation process or mechanism in chicken, especially in the embryonal stage. Thank you very much for watching our presentations.